Hello, we shall discuss on improvement in food production. The conventional means of food production are not sufficient to cater the needs of the growing population. Therefore, plant breeding has become an integral part of uh, food production to provide sufficient food to the ever-increasing population. Plant breeding is a technique of improvement of important crop plants and production of new crops that are better suited for the cultivation, better yield, and even disease resistance. The main aim of plant breeding is to increase the yield of the plant, to improve the quality of the crop, uh, to provide better adaptation to new areas and climates, and resistance to diseases and pests, and even resistance to the environmental factors. Now, the main steps in plant breeding are: the first one is collection of variability. Variability here refers to varieties of plant. In this collection of variability steps, plant having different genes varieties are collected and are collected for breeding experiment. The entire collection of plant having diverse genes in a crop field is called germplasm collection. This forms the first step in the systematic breeding program. Collection of germplasm may be done within the country or from other countries as well. Once the seed is selected, the seed is stored at low temperature ranging from -10 degrees Celsius to -20 degrees Celsius under anaerobic condition. Or the seed can also be stored under aerobic humid condition at room temperature for a short duration. The second step of plant breeding is the evaluation of the selection of the parent plant. Now, in this step, once the plants of different gene varieties are collected, evaluation and selection are done. The plant seeds are evaluated for their genetic compositions, and the desired varieties are selected for breedings. And selection involves picking up of better plants out of the entire collected plants. The selected plants are then separated from the inferior or the undesired ones, and reproduction are undertaken under controlled condition. So these selected plants form the parent plant to start the plant breeding program. The third step of plant breeding is the cross hybridization among selected plants. Hybridization is the technique of introducing characters of two desirable species from two different plants into a single hybrid. This hybridization is carried out by artificial pollination to create new genetic combinations, and it is the mixing of two different lines differing in their genetic composition. A line here represents a group of individual or plant that is descended from the same or related plants. The individuals or lines that is used for hybridization are called as the parent. Now, for example, crossing of two plants, one with high yield in protein and other with disease resistance quality, will result in the formation of hybrid having both the character that is high yield of protein and disease resistance. So these characters are being passed down from two different parents to a single hybrid. So here the two plants having two different characters are referred to as parent, and the offspring that acquired both the characters, that is high yield protein and disease resistance, is called as the hybrid. This means that the hybrid will contain both the genetic characters of both the hybridizing parent. Now, the fourth step is the selection and testing of superior recombinants. In this stage, selection of desired plants from among the hybrid that is arising from the hybridizing of parent plants is carried out, and selfing, that is self-hybridization, is done for several generations to produce uniform homozygous inbreed plants. Inbreed here refers to the offspring that arises from crossing of the closely related parents or from same genetical composing parents. These inbreed lines are crossed again to produce uniform F1 hybrid population. Now in this F1 hybrid, population, this F1 hybrid population can be grown directly as it will contain the desired characters of both the hybridizing plant. The fifth and the last step of plant breeding program is the testing, release and commercialization of new cultivars. The newly developed hybrid varieties of plants are cultivated in the research field and are evaluated for the yield, the quality, nutritional content, and even for their resistance to diseases and pests. The new varieties developed are tested in the fields for at least 3 growing seasons at different locations in different climatic conditions, and only those hybrid that shows their desired characters will be selected and will be released for commercialization purposes. So this is all about plant breeding technique. Now we'll discuss on hybridization. What is hybridization? Hybridization refers to mixing of two different characters from two different plants to one hybrid offspring. The procedure of hybridization involves, the first step is selection and isolation of the plants. In this, the plants that is to be used for hybridization, that is male and female, are selected, and these plants are induced to flower at the same time, and crossing is done in the greenhouse under controlled condition. The second step is the emasculation. Emasculation refers to removal of the male flower, that is the stamen, during the bud stage. So this process of removal of male flower is called as emasculation. Now emasculation is usually done in the bisexual flower. The third procedure of hybridization is the bagging. Bagging refers to the covering of the female flower with a polythene bag or paper bags, so that no foreign pollen may force into the stigma to pollinate the flower. The fourth stage is the tagging. Tagging here refers to the writing of steps, even date and time, in the emasculated and the bagged male and female flower. So this is referred to as tagging. The fifth procedure is the pollination. In this stage, the emasculated male or the stamen is dusted over the female bag flower. The bag is removed from the flower, and the pollen grain is dusted over the stigma of the female flower to induce pollination. In certain crops, male sterile plants have been recorded in many crop plants like maize, wheat, sorghum, carrots, cucumber, etc. In this plant, the male sterile are used as female parents and are crossed by pollen grain of the male parents with desirable characters. This is done to avoid time-consuming process of emasculation and bagging. The main aims of hybridizations are to transfer one or few qualitative characters and use of F1 as hybrid variety. Now there are certain disadvantages of hybridization. The first one is that the pollen grains from the desirable plant chosen as male have to be collected and placed on the stigma of the flowers selected as the female plant. The second is that it is not necessary that the hybrids do combine with the desirable characters. Usually only one or few hundreds to 1,000 cases shows the desirable combination. So even after carrying out the tedious process of hybridization, these are the following disadvantages that are observed in hybridization.